Hello, welcome to the meeting. We're on a different day today, so maybe no one will show up. It's not clear. I didn't get any messages from people, but we do have a lot of things to talk about. We missed last week because of the Labor Day holiday in North America, so I wanted to go this week and do something, talk about some issues. And if you're watching on YouTube, you can go through to different points in the video and just check out some of these features, and I'll try to maybe bookmark them in our Slack channel on DevoWorm, in the DevoWorm Slack channel and the OpenWorm team. So why don't we get started? Maybe someone will show up, maybe not. Let's see. Let me share my screen. Hope everyone's doing well today. Okay. So the first thing I wanted to talk about is this upcoming Neuromatch conference. So some of you have been involved in the Neuromatch Academy this last summer and the summer before, and some of you have also been involved in the Neuromatch Conference, and especially the last one, which was Neuromatch 3. And this is something that arose in response to COVID, but is also a very good idea from the standpoint of having a nice all-purpose neuroscience-oriented work conference. And so they're going to have another conference. I don't know if they have, let's see, this is actually the NMC here. So this link is to the Neuromatch Conference, and this is, so their goals are, of course, simplifying conferences, reducing meeting and travel costs, reducing carbon emissions, and finding new ideas or collaborators. The reason they call it Neuromatch is because they originally had a matching system where people would register, they'd put in some abstracts from their papers, and then there would be an algorithm that would match them with collaborators. But that part has kind of fallen by the wayside in favor of some of the more traditional work conference or workshop setting interactions that you can make. But still, it's a very good experience. So this is the fourth version of Neuromatch. They'll feature traditional talks, lecture-style keynotes, tutorials, flash talks, and posters. And they encourage local meetups during the conference, which is something that actually it would be nice if we could exploit this. It'd be nice if we could have like a developmental session that people could attend, and just kind of like a drop-in, or maybe we could schedule some content during that time. It's coming up fairly soon, so that might not be something we can pull off, but who knows? If people are interested, let me know. So that might be good if we had a local maybe like not a local, I think what they mean is like locally in your community, but we could also have a what they call a satellite meeting, which is where you have a group of people who are interested in a topic, get together and discuss it, give informal talks or things like that. So we could have a DevoWorm satellite for Neuromatch. That might be good. You know, I don't know. Again, I want to gauge interest on this. I don't want to force people into it, but I also want to be able to do it if we can. So the registration date for the main conference is pretty flexible. It's open in October. Register before November 30th. I think critically, though, the submission date is October 15th. So let's keep that date and write it down, and make sure that we kind of set our minds, if we want to submit something, set our minds to that date. I think we have a number of people in the group who have done excellent work that would match the criterion of this conference. They're interested in computational neuroscience, so they're interested in traditional neuroscience, but also in computational neuroscience, and then a lot of deep learning, machine learning, and so forth. But this, this kind of work would have to be a little bit more connected to either theory or the brain. So I don't want to just have you think that they usually don't reject submissions, but typically people don't present things on like a machine learning analysis. It's usually something a little bit more like what we do in this group. And I think, maybe some of the work we've done, maybe even the DevoLearn stuff would be good for this. So I would encourage people to make their submissions, talk about it with me, maybe present in the group, and then, this will be a good experience for you to put together a talk and give a talk in a session. A main conference then is from December 4th to 6th, so it's towards the end of the year. So all you need for this submission is like an abstract. It's like 250 words. It's very simple. You just, describe your work. Usually it's like the intro, the main body of the main idea of the talk, and then a conclusion, which is usually like future directions, and then that's it. And there's no paper. You don't need to submit a paper for this, so this is pretty flexible. They don't have the agenda yet. Registration fees, you can pay $15 or pay as you wish after the registration. So it's like they're not going to hunt you down for money if you can't afford it. And then the local meetups, of course, they're calling for that. So that's something to keep on the calendar. Another thing I'd like to bring up here, and this is my other group, and I'd like to talk about this a little bit. This is the Orthogonal Research and Education Laboratory group. And this is something that I host as the head scientist, and I work a lot with Jesse Parent, and I think a number of other people from this group have also gone over to that group and done things. So we're a pretty flexible open science group. We do a lot of different types of research, and I bring this up here in this meeting because I want people to kind of get a flavor for research, and in the ORL group you can do it in a lot more broad fashion than in this group. So we have a bunch of opportunities open now. We don't really have any money for this, but the idea here is to gain experience with the research and to engage in research and do other things. It's a very good opportunity for young researchers, especially people who don't have PhDs, non-traditional scholars, people like that, to really kind of get a taste of research and do it at their own speed. We have a lot of people who come and go in the group. They come in, they do some work, and then they leave, and then they may come back and do more work. So it's pretty flexible from that standpoint. But right now we're looking for people who are a little bit more sort of devoted to their work, and they want to do this on a weekly or monthly basis. So we have a couple of opportunities here. We have an advanced opportunity here for a research assistant. This is someone who would work sort of with Jesse on some things, like a lot of lab management activities. So this group, this ORL lab is all about taking initiative. So, we have a lot of things going on, a lot of ideas in our meetings, a lot of ideas in our Slack, and it's up to you to take a piece of that and, work on it, take ownership of it, work it out, and, do presentations, produce scholarship, do the science that's involved in it, all those things. And it really gives you a good opportunity to engage in a research project. So this position is a little bit higher up on the ladder. This isn't just like a research position, like taking ownership of a project. This is actually doing some management activities, and so you would lead or generate your own project with support from existing lab managers. But then also on top of that, doing administrative things. So this could be things like maintaining our medium posts. We have a lab medium, developing educational materials, literature reviews, coordinating efforts to engage people, things like that. So we have a lot of different things that you would do here. You wear a lot of hats, and it's one critical aspect of this is you have to have a real commitment to interdisciplinary research. So it's a very good opportunity to get that up, to learn how to do interdisciplinary research, especially like crossing over from one discipline to another, and seeing how those things combine in a way that's sustainable and that you can actually build projects off of. So and then there are a whole set of opportunities related to project management that you can gain from this kind of position as well. There's model builder, which is a different position. This is I think a lot of people in this group might be interested in this. This is where we have model builders who, you attend a meeting, and you could attend this meeting and do this, where you attend the meeting, you listen to what's going on, and then you take some idea and you model it. And what I mean by model is you want to turn it into something that you can understand with a computational model. So, we're interested in people who have some background in reinforcement learning, genetic algorithms, deep learning networks, complex networks, agent-based models, and other types of models as well. So if you have this background, say you have a background in deep learning, this position might allow you to broaden your skill set and pick problems from the wide range of problems we talk about, and work out some sort of implementation. But then also you have the opportunity to learn other methodologies and then work on those as well. So this is a very good opportunity for this. And then there are these other project-based internships, which are just based on our projects. Some of the projects are far along and some aren't. The AI ethics, of course, a couple of people in this group have done this. We have something called Cognition Futures. So this is, if you want to find out more about this, contact myself or Jesse Parent, and we can put you in proper, I think we're doing a lot of the evaluation, so you would be contacting the right people for that. Then we have an administrative position, which is an assistant lab manager or social media manager. This is more directly just like administrative stuff, and we're already kind of reviewing applications for this. This probably wouldn't be very good for people in this group for the most part, but I just wanted to point out some of these opportunities. And this is it's off of our main orthogonal lab, orthogonal research lab website. So this is this website here, orthogonalresearchweebly.com. This is our main website, and you can get there by going down to people, get involved, and then going into this open positions. So that's all I wanted to say about that. Hello, Ujjwal. How are you? Looks like Ujjwal's here. He's muted. Okay. Hello, Ujjwal. I'll just say hi. You can just watch what we've got going on. So I'm just trying to update people on things this week. Next, I wanted to talk about our Hacktoberfest activities. And so we have Hacktoberfest coming up. So Hacktoberfest, we've talked about this before. This is this activity that is sponsored by GitHub and some other organizations. And the idea is to take the month of October, and the Hacktoberfest moniker comes from Oktoberfest, which is a German holiday during October, and they apply it to just doing stuff on GitHub during that month. So the idea is that you have these organizations that participate, and they usually open up a repository, or maybe a couple of repositories for public contributions. And the idea is that people contribute during Hacktoberfest, and if they make 5 or more contributions, they get some swag or something. So I don't know if we ever really have any swag to give. I'll have to figure this out. But definitely, it's something that will lead to bigger and better things for people. There's definitely going to be some social media recognition for people who participate. I'll prepare a blog post so people will be recognized. So this is a big event for open source enthusiasts, held every October. This year it's from the 1st to 31st of October. It's organized by DigitalOcean along with other sponsoring companies. So every organization has their own sort of specific Hacktoberfest organization and their rules. So, we want to do this. We have to start planning this out now, which is why I bring it up today. And I know that I think it was Krishna and I think Mainak who talked about this last time we had a meeting. And so I don't know if what their interest level is in this. We definitely are accepting, we're always accepting contributions to DevoLearn. We're also accepting contributions to some of our other repos, but DevoLearn might make the most sense in terms of focus this year. So this is we just have to come up with some of these different things here. We need to come up with, I need to come up with a social media post which would lay out some of these rules. So I guess the rules are just kind of like, what do you need to do to be recognized? And what I did last year, actually, we didn't, well, I guess we did formally participate in it last year. My rule was like, if you make one pull request and it's merged, then you get recognition. And then Mayuk Deb has been working with people to make sure that their submissions are successfully pulled. So thank you, Mayuk, for that. So people will get at least one, probably, unless it's just egregious. But still, and if you get one pull request accepted, then you usually get some recognition. It would be nice, though, if I could get some sort of like swag for people, but that's not something that, it's kind of one of those things that I don't know how motivating that is. But anyways, this is a pretty successful effort. So if we want to do this, what I think I'll do, I will make like some general rules for it, and then I'll post on this. We'll start out with DevoLearn. We'll assume that that's going to be the one we're going to do, and then if people want to have other repositories included, we can just simply get those in shape and ready for submissions. And so I think the only thing we really need to do is advertise where to contribute, and then have a badge in the README of the repo, and then have someone who will go in and make sure that these submissions are accepted or guided properly so that they can be accepted. And I think the rule of just having one merged pull request is good, since we're not really giving out swag. If someone contributes like 15 really good pull requests that are merged, maybe we'll give them some sort of swag. I don't know what but so we'll make the rules. We'll figure out what I'll kind of work through the rules, and maybe next week I'll show them to the group. And then this is just more about like how this organization is running their Hacktoberfest. But I think this is a good opportunity to get new people into the organization. I think when you have when we have GSOC, and then we also have a Hacktoberfest. Although Hacktoberfest isn't really self-sustaining like GSOC, it's still an opportunity for people to come in and contribute. So that's good. Now, speaking of DevoLearn, I want to go over some of the different things that are going on with that. So as I said, thank you to Mayuk and Mainak Deb for being maintainers of DevoLearn. Now that GSOC is over, we're going to be working on a paper on this, but we're also going to be continue to accept pull requests. And so we have one pull request open now, and we had one a while back, and I see that we're getting some new contributors here. So Somnath Sharma is submitting a pull request today for setting up CodeCov, which is an issue number 56. We had another pull request the other day from another contributor I had not seen before. So it's good that we're getting ever more pull requests. That means that this project, is still going pretty strong. We have a number of different issues here, 6 issues that you can address. So these will be, maybe we'll add a couple more for in time for Hacktoberfest. In the next weekly email I'll send out, I'll make a call for, sort of coming up with different things that we can put on. If we have a repo open for Hacktoberfest contributions, then we should probably have a set of issues that people can address, because that's the easiest way to do it. People can, check out the issues board, find an issue, do something, make a pull request, get it accepted, and then they get their recognition. That'd be a pretty easy pipeline to follow, I think. So that's the way we'll do this. And we can do this in any of the other repos, it's fine. Yeah, so that's DevoLearn. Then I'll go to major tasks. We haven't talked about this in a while. I think we went over a couple weeks ago, but it's getting kind of unwieldy here. I was trying to figure out where we are exactly on this. I've been trying to update it accordingly, but I come up with ideas for things. We come up with ideas for things in our meetings, and I put them on the board and kind of break it down to a couple issues, and then I kind of come up with another issue that's maybe related. I really wish there was one function I wish that this board had would be to link issues together sort of graphically, so I could see how they're all related, but because I can't remember this very well, but we'll go through. So first thing we have is this off the radar set of issues, and these are things that we've mentioned but we've not really touched very much, and then they're gonna get forgotten. But I don't want to really forget about them, because some of them are actually pretty decent ideas, and maybe it just takes someone to come back and revisit it to get it to happen. So I think actually this Krishna paper review is finished. Alright, so we have this CT computed tomography via cloud computing. We have this update DevoWorm ML lectures. This is something that's kind of fallen off my radar because I was doing that earlier this year, but I just haven't had time recently. This issue 30, follow-up on neural organoids. So we talk a lot about organoids in the group, but we haven't really done anything scientific with them. And I've been talking for like 2 years with people about how to do sort of organoid data science. And that is can we take secondary data sets and analyze them and bring out insight from these data sets. A lot of the experimental data sets with organoids are, people grow organoids in a culture, they put in some fluorescent markers, they look at the organoid itself, sometimes they do some cell tracking, but most of the time it's looking at different physiological functions and identifying gene expression. So there really is an open area here for other methods, or for taking those data and finding, constructing measures and doing some other types, answering other types of questions. The first step in this, though, is to create or to go to the open data set repositories and extract the data from there. And we have this, we've done this with C. elegans, we've done this with sea squirts, we've done this with Drosophila and zebrafish. So the data exist. They're not as prevalent as some of the model organisms, but it's still out there. And so we can follow the same procedure to do something with organoids. There's this Lagrangian embryo, which is sort of a mathematical model for an embryo that's evolving and dynamically. And there's a Lagrangian, which is a dynamical function that one can use to characterize the embryo. This is something that I think Dick Gordon presented on a long time ago, and if you're interested in that, we can revisit it. It's an interesting concept, but it was never really developed very much. We have this review compositionality repository. This is related to some of the stuff we're doing on game theory and on category theory. And again, this is something that's been just kind of not really developed. Models of Biological Living Networks paper. So this is based on our embryo networks work. This is just something that is a next step in that direction. There's been some progress on this. I don't know where to put this. It could be an action item because we've done some presentations this year for these networks and how to model networks, not as sort of static entities, but entities where you get new cells coming into the network. So you get new nodes and you're growing the network and you're gaining connections, but the rules of connectivity aren't like most other networks because they don't behave like most other networks. Like if I'm talking about social networks or telecommunication networks or power grid networks, things like that, they have a different behavioral mode and we don't really know what that is. So this paper would sort of lay out what exactly those rules would be, what kind of net. It's a special, developmental networks are a very special kind of network. They're not like the other complex networks that we know. And if you know anything about complex networks, you know the terms like preferential connections, the terms like scale-free and small world. Those are the kinds of things that people try to use to characterize these different types of complex networks. But biological networks might be very different from that, especially like embryo networks or developmental networks where you're adding in nodes. And there's a spatial aspect to it that's very different from other types of networks. So that's something that I'll actually put in the action items section, because I think it's, probably something we can do in the next several months. This lecture on the artificial embryo, that's something I was working on, like a numeric embryo. I don't know about a lecture, but this work has been incorporated into a couple pieces of work, like for example, into the paper we did for biosystems. And it was published at the beginning of the year. I worked on a numeric embryo for that. So some simulations of embryogenesis, the process, simulations of a lineage tree and that sort of thing. So I haven't actually put a lecture together on that though yet. So that's off the radar, but maybe that's an action item as well. So we have these things that are finished. That's good. It's a long list. It's better than a short list. Very long list, actually. We've done very well on that. In progress, we have a number of things. We have this axolotl embryo animations and segmentation. We're working with Susan to get data. We're working with Akshay and maybe some other people to work on the axolotl atlas, or this three-dimensional sphere that you can map these images onto, so you can explore the surface of the embryo. And this is something that's definitely in progress. This analysis of shapes of polygonal archaea and shape droplets. So this is analyzing cell shapes. This is something I think my NOC is engaged with. This Bacillaria non-neuronal cognition paper. We'll talk about that in a bit. And then that's all for the in progress. The action items are maybe a little bit less in progress than in progress, but we have a number of things here. We have this networks paper, updating the website, which is actually sort of a continual thing. Revising the ANNs, BNNs paper. This is the artificial neural networks, biological neural networks paper. And actually I would say that for this issue, and this is, it's Krishna, but it's maybe also Jesse and myself and maybe a couple other people. There is actually something, maybe we'll submit a follow-up talk to the NM4. NMC4, I guess it's called. Okay. And then, so we did this, we talked about this last year at NM3. which was last October or November, I believe. So this is actually something we can follow up on. I think the talk would have to be like, we could take the original talk and modify the slides a bit based on what we've learned over the past year. So in the past year, we've written a paper on this, a preprint. We've also done some additional work in the meetings on discussing, this link, and there's been a lot of stuff published on the link between artificial and biological neural networks. So I think an update on this would be good. And then we'd revise the paper accordingly. This early life and origins of development theme, this is something we talk about a lot with respect to the early evolution of embryos, which are, have some common ancestry in an organism that needed to have an embryonic stage of development. So, this is thinking about really early Earth, really early life, and the conditions that led to maybe like the development of embryos, or what a common ancestral embryo would look like. This is interesting. This is a little bit speculative. We don't have a lot of data for this, but it's nevertheless something we can, if someone wants to grab onto it, you can. More issues related to the axolotl data. And, just some follow-up items. It's mathematics at DevoWorm, which we're still following up on. This complexity measures, which has never really been a strong project, but it's been a theme. There are a number of things in the meetings that we talk about all the time with respect to complexity theory and, different things where we have an open question of, like, we have this complex process. How do we put numbers on it? How do we put, like, measurements on it? Can we build a simulation around it? And if so, how do we do this? So this is what complexity measures is, and it's a pretty broad theme. It's sort of infused in a lot of what we do, but it's not like something that we've really focused on, cataloging. And I wanted to point out that this mathematics at DevoWorm kind of fits into, these are kind of companion issues, even though one's number 3 and one's number 89. I think we can advance the mathematics at DevoWorm by sort of thinking about this in terms of complexity measures and vice versa. So that's something I want to put a sort of a pin in for that. Other than that, we have these to-do and hold items, and these are just things that hold is like, maybe it's not quite off the radar, but it's, don't really have a, we're not really pursuing it right now, but we're still thinking about it. Like tutorials for YouTube, or this morphogenesis and deep learning paper, creating an embryo model in Blender, these sorts of things that are sort of, we perennially talk about them, and so these are things that we want to kind of focus on. I think this is going to be off the radar, actually. Okay, so that's what those are. And then these to-do things, these are things that kind of we thought about in meetings, and I put them in here, and they haven't really found their way onto the list. I think this goes here, but this is this Kindle book idea. But then there are other things here, like the virtual developmental worlds. I think we talked about that last week, and that relates to these issues here, number 38 and number 6. We're trying to create a visualization for a Docker container, but also for more general outreach, and for just, one of the things that we can offer to the public. So these are all things that are to do. Now, if you want to update this list, if you want to create issues, or if you want to address issues, go to the DevoWorm GitHub repository, or the DevoWorm organization. I think it's actually not an organization, but you'll find github.com/DevoWorm. Go to the group meetings repository, and then go to the projects. And this is a project, the project board major tasks for 2020-2021. And just, if you have permissions, you don't have permissions, let me know. Request permissions, and I can let you in. Or you can send me a message in GitHub, or via email, or however that you want to add an issue, and I can add it in. So there are a lot of ways you can do this. Okay. So we'll move on then. I just wanted to assess where we were. We don't really have, we have a submissions document, but we really don't have anything on that right now. The submissions document is just kind of like, you know, different conference activities, publications. We're no farther along in anything this week than we were last time. I think we did have something on, well, we did have the dynamics days, but that was finished. So, hello Akshay, how are you?
Oh, it's just you?
Yeah.
I'm sorry,
yeah.
I'm
It's okay.
I have some network issues. I don't know when it will disconnect.
Okay.
I'm just trying. Yeah.
Well, welcome.
No joy.
Yeah. So yeah, I'm just reviewing some things. If did you have any news or you want to share with us?
Actually, no. I haven't made any progress because my exams were starting, so couldn't work much. The work you didn't actually message me on Slack, so I was actually waiting for that.
Oh, about
There was a work we discussed. Yeah.
Well, I didn't know exactly.
About the math.
Yeah, I didn't know exactly what we're going to do with that yet. Still, I'm going to actually talk about that right now, and then
it's okay.
Yeah. Okay, I'm sorry. I'm sorry I didn't get back with you, but it was yeah. So
no. It's fine. I just wanted to check because I uninstalled Slack, and if some workspace change or something, I don't know. That's all. So
yeah. Okay. Well, glad you're doing well. I hope you're doing well on your exams, or did well on your exams.
Oh, it's getting too tough. Like this is my third year, so I have one more year after third year. I mean, it's just 4 years course, so after that I'm gonna be like done with my undergraduate studies.
Oh, good.
So
yeah. Well, that's good. Yeah. So then I'll actually I was gonna get into the Bacillaria stuff now. So let me share my screen again.
Yeah, sure.
Yeah.
You go ahead. Yeah.
Okay. So this is the Bacillaria paper again. We're pretty close to the end. I wasn't sure. Yeah, so that needs to be like 12,000 words, I guess. I wasn't sure exactly what the next steps were on this, and I think I might wait until I go ahead and submit it to the people who want it, and then we can see what they request. So there's a lot of stuff in this paper on different methods. So it's sort of a cornucopia of methods. The idea basically is that there's this organism Bacillaria. It's a diatom, which means that it has sort of like an algae, but it also has the properties like it can move around. It has a lot of motility, and there are a lot of interesting behaviors that they perform. So there are a lot of different types of diatoms. There are round diatoms and these long rod-like diatoms. And the long rod-like diatoms are the ones that we're talking about here. So they're eukaryotic algae that have chlorophyll and silicate cell walls. So the cell walls are very rigid, but they can also move around in their environment. And so this is, it's very small. It's like 80 microns by 10 microns, which is very small. You need a microscope to see that. They're even smaller than C. elegans. And there's this whole sort of, you can see these colonies here, a bunch of cells that are sort of put together by these secreted polymers that allow them to stick together. And so they slide against one another like an accordion. And so that's the main mode of movement we're interested in. But there are also other things that they do, like they extend out and they hold there, and they will break apart over time, so they age. So there's a lot of things going on here. We were going to use some data to fill in the gaps here. We were going to do we did a paper 2 years ago that was on a formal data analysis of the movement. And so that was pretty well received. It was just published as a book chapter recently. So we have the data set. We have like a data set of movement. We have some analyses. deep learning and biomechanics, but this is more speculative, and this is like, talking about maybe the potential for what they call non-neuronal cognition. So non-neuronal cognition is this idea that these diatoms will behave in a way that's sort of reminiscent of a cognitive processing type set of behaviors. So they don't have a brain, obviously. They just have their own physiology, and they generate thrust, and they interact with one another. But the idea is that they have this sort of collective behavior, and they have this sort of like they can take in sensory information and respond to it. So it's in that way, it's kind of like a psychophysical nature of cognition. And so a lot of this is, we talk about a lot of different models for this in the paper. We talk about different psychophysical models. We talk about different concepts in neuroscience that might apply to some of the things going on here. And then we have this model of a CoPG, or a collective pattern generator, which is like, there's this idea in, actually in C. elegans, you find these a lot. They're central pattern generators, and they're generated by networks of neurons. So the neurons, let me actually go to my board here. So a central pattern generator is like a series of neurons that, I don't know if I can get this in here, okay, it's not working, so we're not going to get that. Let me see if I could replug it in. Okay, there we go. All right, so a central pattern generator is like a network of neurons. We'll just take two neurons here as an example, and these two neurons will generate pulses in a way that creates an oscillation. So the oscillation will be like a sine wave like this. All right. So the idea is that this neuron fires, then this neuron fires, and this neuron fires, and it gives you this oscillation, and this generates a collective pattern. There might be a bigger network of neurons. You might have like add a third or fourth neuron in, and you have these networks that are synchronized so that they either fire in sequence or they fire together, and they create these sine waves or some sort of pattern that allows you to generate this rhythmic motion. Sometimes you'll see these in generating movements like swimming movements, or like limb patterns, like in some amphibians. They're in their spinal cords, they'll have these central pattern generators that generate their legs moving back and forth, so they're generating like locomotion. There are some organisms that have these in their spinal cord where they're generating tail or fin movements that oscillate. So all these things are generally oscillatory. And so you can see that in the Bacillaria, these colonies also, if you look at the first paper we did, they also have this oscillatory pattern of movement. And so, but then in the case of the Bacillaria colonies, you have these rapid-like cells that don't have any neural activity. They don't have any, they maybe have some limited electrical activity, but it's certainly not like an action potential that you'd see in the neurons up here. So, this is something that is generated by these cells moving against one another. It's like you have two cells that move against one another, and then these two cells and these two cells and so forth. And this sort of coordination propagates across a colony, and it produces this oscillatory behavior. And the idea is that these two things have a parallel. The parallel is the movement output and some of the activities within the colony versus in the neural network. So we have a special class of pattern generator called a collective pattern generator, which is something we talk about in the paper. So that's what we're doing in this paper. It's a very it kind of hits upon a number of different models, and kind of this idea of aneural cognition. People have published on it in different organisms. So like in paramecium, or in the water flea, or in Physarum or slime molds, even in like protocells, which are these simple bags of chemicals, bags of like RNAs and other things, lipids, and oil droplets even, which are not even organic. they can break their symmetry when exposed to water. So this is something that you see across a lot of different systems that don't have nervous systems. But it's nevertheless, they exhibit this sort of cognition. And so one of the problems with aneural cognition is it's very much an analogy. It's like, okay, it's a nice analogy, but people anthropomorphize things all the time. They say they behave like humans, and, obviously they're, like paramecia don't behave like humans. They don't have the same brains, they don't have intentions and motivations like humans. At least we don't think so. We can't really show that definitively, but we can use these, these analogies. But then that means that the power of your model is weak because an analogy isn't particularly strong as an explanation. So we bring this sort of approach to bear, and we bring up a lot of different things here, a lot of different psychophysical mechanisms. I think if, in Akshay's case, if he's interested in doing some modeling on this, I would just have him read this paper. Like I'm going to submit it, and then I'll have, Akshay, I'll have you read the paper, and then, maybe think about like where you could contribute in terms of a model. Like there are a lot of things here. There's like heavy in intelligence. There's like some simple psychophysical equations. There's like the free energy principles. So there are a lot of things that we throw around in here that are not like,
okay.
Yeah, so it's kind of hard to say like build a model because there's so many things in your connectionist networks. So yeah, I mean, I'll send you a copy of the paper. You can read through it and
yes, I'll do that. You could submit this in chat, or personally you're gonna send?
We'll probably like have it, put it on. I'm gonna send it out for review in the next few weeks. But we're probably gonna have like people say, well, you need something more. And so that's where it would be. Yeah, and so that's where it would come in handy. By the second round of revisions, I think we maybe have something. And if we don't put it in the main paper, we can still have like, you could be an author. You could find a way for you to contribute to it without, but then the model, we'd have it available. So like, if you've ever seen papers where they have a paper and then they have some supplemental model online somewhere, that would be something we could do too.
Yeah, that sounds good. Yeah.
Okay.
You share the copy, I'll go through that, then I'll get back to you if I have some questions or anything.
All right. Yeah, I'll make a note of it.
Okay.
All right. Very good. So that's all I needed to say about that for now. Hopefully this will be done in the next few weeks, because this has been one of these things that's been a grind to get through, trying to get everything together. But we're getting close. We have a lot of good references in here as well. Yeah. So earlier we were talking about Hacktoberfest, and just as an update for Akshay, I'm going to make up some rules for our Hacktoberfest here in the group, and then next week maybe we'll go over in more detail what's going to be part of it. So that should be good.
Okay.
Yeah. So then finally I wanted to get to our papers. So I've got a lot of things here. I'm just going to go over a couple things. And I know no one like Akshay and I are the only people here, but I'll go through some of these things.
Shruti came. Shruti actually joined.
Oh. Hi, Shruti. How are you?
Hey, I am good. How are you?
All right.
I just realized that the meeting was scheduled today. I was just checking my mails. I thought, why not drop in?
Well, welcome.
So how are you?
I'm doing all right. Yeah. I just couldn't make it yesterday. I had another engagement, so.
Oh yeah, I didn't realize that. So just
Okay. Well, I'm going to go through some papers and
All right.
And we'll be you can watch the earlier parts on YouTube. There's some things that I mentioned about Neuromatch 4 coming up, and then also the Hacktoberfest stuff, which is happening.
Oh yeah, Hacktoberfest 2 will be coming in a few days.
Yeah. So our papers are, so I want to go over this thing. This is something that is, these are simulation notes that I don't know if they're ever going to happen, but I just wanted to go over these with the group, see what they thought. This is something that, so I've been showing this mathematics of DevoWorm thing last couple meetings, and it's not a paper, but this is like something that the group has been working on towards a realistic biophysical simulation of C. elegans, and it hasn't really been updated in a long time. We don't really have it as a preprint, but this is something that gives you an idea of what is going on with the OpenWorm project. So the idea is that these are the biophysical foundations. Basically what they're trying to do in the broader OpenWorm project is they're trying to simulate the movement and the nervous system and the coupling between the nervous system and movement in adult worms. So our goal is a little bit different in this group. We're interested in development, and so development requires a whole bunch of other models because, you know, you have like nervous system models, you have movement models, but development's quite a different process than that. It's like, how does that thing get there? And so we have a lot of different models that we use that are different than this. But what they're doing here is they're laying out a lot of these models for people to read about. There's software, there's simulation. You can see it do cool things, but you need to know the mathematics or the theory behind a lot of this. So this wasn't, well, I guess this is maybe a finished draft, but like the idea is that you have, you know, a couple of components. You have the basic equations of motion, so you have nervous system simulation. There are a couple different types of models that they use, and this is just like, you know, where you're simulating neurons and you're saying that these cells have different modes of generating action potentials, which is the way they communicate with one another. So there's like the integrate and fire model, there's a single compartment model, there's the multi-compartment model. And so these are ways you can model neural activity or how to generate action potentials within the cell. And, you know, because you don't know how, for example, you know, you might have a very realistic model with all the parts that you need, but you don't know how it actually does its, how it actually proceeds with its process, so to speak. And so these are actually from the literature on, in computational neuroscience, where people have like proposed ways in which neurons do this in an efficient manner. So they cover these models. Then we move on to the smooth particle hydrodynamic simulations. So if you've seen the stuff that Cybernetic has done, it's a group within OpenWorm. They have these biophysical models where they have like a worm on a surface, and the surface could be like a gel or something. And they simulate the movement against that surface. And they also look at hydrodynamics, where the worm is in a liquid environment, which it usually is. And so they use these particle simulations that have like hydrodynamic properties. So they have like different drag forces, and the worm is generating forces against this, and it's sort of, gives you a model of what is sort of the path of least resistance for the worm. So, why does the worm move in certain ways? Or C. elegans has a couple of modes of movement. Are those like the most efficient modes of movement given its physical constraints? Or, that's what we can figure out with this. And so there are a couple of things to lay out there, different algorithms, and then extending this to modeling biological tissues. So they pick an algorithm that they think is effective, the smooth particle hydrodynamic algorithm, and then they apply it to the model, and then they get results. And this just doesn't worry about the results, it just lays out the method. And then this Boyle-Cohen model of neuromechanical coupling, which is published in the literature. This is Netta-Cohen and Boyle. This is electrical properties of the muscle body, muscle arms, and coupling. This is all the components of this model of basically coupling the nervous system with movement and mechanics of movement. So this is the mechanics of movement here, where the body is moving through a medium. this is the nervous system, and then this is sort of integrating that. So this is a nice set of models, and then putting it all together, which is a page which is kind of pulling together all of these models into one simulation environment, and then references. So, you know, this is just like, this is laid out like a math paper almost, where you have the methods, you have the equations, you have descriptions of the equations here. This is incomplete, so it's not, and then you have an algorithm listed here, so you have like this, and yeah, I mean, this isn't really complete, but this is the idea behind a lot of the what OpenWorm is doing, and we have our own sort of methods. So I think we can do something very similar to this for DevoWorm, and maybe even put it into this, although maybe it serves as its own little document. So I just wanted to show that for, you know, thinking about this mathematics at DevoWorm and where that would go. But also, this is very good if you really want to understand what's going on in OpenWorm, because I think there, you know, there isn't a lot of like transparency there in terms of like, if someone wanted to know, you know, how do you know that a worm neuron does this? Well, we parameterize it with data, but we also use these models to sort of make predictions about what the worm is going to do. So, let's see. I also wanted to talk about, I think I'm going to talk about this paper here. So this is a paper on network science. We talked about network science in meetings prior to this. This is something called a universal route to explosive phenomena. And this is a pretty heavy-duty sort of network science physics paper, so I don't want to really get deeply into it, but I'll talk about it a little bit. So the abstract reads, critical transitions are observed in many complex systems. This includes the onset of synchronization in a network of coupled oscillators, or the emergence of an epidemic state within a population. So we talked about coupled oscillators before. They're these neurons that are connected in a network. So these two neurons are coupled. They have this internal process that generates action potentials. Then there's this output, which is, you know, it's like a rhythmic, you know, regular pattern, and that gives us this, you know, so you can synchronize these oscillators are coupled in this way to generate this patterned output. Or the emergence of an epidemic state. And of course, we know from the pandemic that when there's a lot of transmission of a virus, you can end up, if the transmission rate is high enough, with an epidemic or even a pandemic where you can't control the spread of things and they remain in the population, you can't knock them down. So there's this sort of explosive first-order transition that, which they mean by, what they mean is going from like nothing to this explosion of activity and this sort of order, you know, so it's like explosive in the sense that it just appears. This is a first-order phase transition, meaning it goes from one state to another very suddenly and in a nonlinear fashion. It's hard to know like when these things will happen in a system. They just happen very suddenly. This has caught the particular attention of investigators of a wide variety of systems when classical models are generalized by incorporating additional effects. Here we give a mathematical argument that the emergence of these first-order transitions is not surprising, but rather universally expected. So varying a classical model along a generic two-parameter family must lead to a change of the criticality. To illustrate our framework, we give three explicit examples of the effect in distinct physical systems. So the systems are a model of adaptive epidemic dynamics, for a generalization of the Kurmoto model, and for a percolation transition. So I think epidemic dynamics we know is, you know, like maybe the spread of a disease and how that changes with as the virus adapts to the population. The Kurmoto model is actually a model of things that generate these sinusoids, like I showed you in this image. So there could be neurons that fire and generate sinusoidal output. It actually could be fireflies. There's a model of fireflies where if you go out and you look at fireflies, like a bunch of them sitting above the grass or whatever, you'll see that they synchronize their flashes. So they have the sac of luciferase in their tail, and it flashes as they they're coordinating their behavior. They're communicating with one another, and over time, those oscillators, as this sac starts to glow and turn off, all of the fireflies in a certain area will synchronize their flashing. And so this is something that happens sort of, it's hard to say that they coordinate it closely. It just happens over time with some limited interactions. And then percolation is where you have something that runs through another medium, like water running through rocks. If you've percolated coffee, you know that the water, goes through a bunch of coffee grounds, and if it's hot water, it'll, go through the coffee grounds and you'll get coffee at the other end, but it has to percolate through this medium of grounds in order to do that. And so percolation transitions are where you have this process, but there's a transition in the state of the material where the water is going through. So if you have coffee grounds at the beginning of your coffee, it's very loose and grainy, but then when you're done, after you've run the water through it, it's very sticky, and it's, sort of almost like a solid. And so this is this percolation transition, where water going through it will change the sort of this physical state of it. So they say that this is a general property of a lot of systems. So this is a lot of statistical mechanics. I'm not going to get into this, but there are these, so you have what they call critical order disorder transitions as a system parameter is varied. And generally speaking, if you vary the parameters of a system, so if you vary the parameters of some system amongst the fireflies as they're synchronizing, they're flashing, or you vary the parameters of water as it's going through coffee, you can get these kind of different transitions. In statistical mechanics, that could be a continuous or discontinuous transition order change at the transition point. We're talking about first order phase transitions, which is the thing that people are really excited about in complex systems research, because they lead to this explosive change of systems properties. And so for a wide variety of complex systems, it's been observed that a variation of the model by additional features leads to the change from a continuous second order to a discontinuous first order phase transition. So the second order is continuous, where there's no explosion, it's just kind of like gradual. The discontinuous or first order is where there's some sort of step function. There's a change that happens very rapidly and just suddenly, and it's hard to predict. So that's that. And then there's some equations here that kind of describe. They're using ordinary differential equations to describe these sorts of systems and how this change happens. Basically, this can capture a phase transition, although they're very hard to predict in advance when they'll actually happen. I don't know if there are a lot of figures in this paper. This kind of shows you this bifurcation process where you have, this discontinuous change in state. So the gray cases are first order subcritical transitions. These black lines are second order supercritical transitions. That just means that this is where you see the most change, the most explosive change in these black line cases. And then this just walks through a lot of this. The reason I picked this paper, I think it does apply. There's some nice developmental applications for this, and we've talked about this off and on in the group, and I don't know, maybe not recently, so maybe it doesn't make a lot of sense to people now, but the idea is, that there are these symmetry breaking events in development. So in an embryo, you'll have symmetry breaking. You also have which is like where you have symmetry, you have a right and a left, you have, things that are undifferentiated, and then suddenly you have this breaking of the symmetry where, say, like two cells, one on the right, one on the left, are different sizes, or one forms a tissue and the other one doesn't, or the tissues are differentiated within the embryo, or there's some difference in the different parts. So there's a symmetry breaking. And this is one of the things, when you see the symmetry breaking, this process can be described through this sort of explosive change. And so they don't really have any figures in this, I don't think. Yeah, so it's a lot of equations. But this is the relevance of it to development. And it's actually quite broadly relevant. And one of the arguments about, one of the criticisms of this approach is, can you take like mathematical equations and apply them to such a broad range of systems and say that that's a universal property? Some people say no. Well, obviously, if you're a statistical mechanics person, you say yes. But this applies, I think, more broadly to machine learning, where we have machine learning algorithms that, we apply to every type of data. And maybe that's not the best strategy. Maybe that's not something that, are we picking up on these universal properties, or are we just kind of applying it broadly and not really describing the system evenly? One system we might describe very well, another system we might not describe it very well. But in their defense, of course, they do differentiate between different types of systems. So they say, well, you can use this on a wide range of systems, natural, mechanical, but it has to have certain properties. And so that's what they're laying out here. Next paper I'm going to talk about is this, towards a physical understanding of developmental patterning. I think this kind of ties in with the other paper. Now, the other paper was hardcore physics. This is more hardcore, I guess. It's in a biology journal, developmental biology journal. Oh, it's in Nature, but it's, it's not physics. So there's this towards a physical understanding of developmental patterning. So the abstract is the temporal coordination of events at cellular and tissue scales is essential for the proper development of organisms, and involves cell intrinsic processes, which means processes within the cell. That could be gene expression, that could be protein synthesis, just things that happen in the cell. And that can be coupled by local cellular signaling and instructed by global signaling. So these processes are coupled by signals either within the cell itself or outside of the cell. So you can think of the cell as a compartment within a larger system, and these compartments interact through the signaling, or they're signaling within a compartment. And so all of these things create spatial patterns of cellular states that change over time. So these compartments exist independently in space. They signal each other, they change, and they differentiate due to these signals and their intrinsic processes. The timing and structure of these patterns depend on how an organism develops. So this determines how its developmental trajectory. If you want to think about a simple model of development, it would be that. It would be these compartments with a larger field, their signals, their processes internal to these cells, and then that's development. If you get different outcomes, it's because you have different signals, you have different things going on inside the cell. So traditional developmental genetic methods have revealed the complex molecular circuits regulating these processes, but are limited in their ability to predict and understand the emergent spatiotemporal dynamics. So now we add another aspect, which is the spatiotemporal dynamics. And you can't really predict that using molecular biology. In other words, if you go into one of those compartments and you probe those endogenous or those internal mechanisms, it doesn't tell you a lot about the broader embryo. It just tells you about what's going on in that cell. So we need to have approaches from physics to capture a lot of these things that are going on across multiple cells. And so this signaling is one aspect, but there's something more to it. There's a physics that is involved in how the signaling gradients are set up and things like that over space and over time. So combined with advances in imaging and computational power, such approaches aim to provide insight into timing and patterning in developmental or developing systems. So they talk about embryos here. They talk about transitions between states. So transitions between cell states can be visualized by changes in gene expression levels, which is regulated through the activity of transcription factors. Now that's quite a bit different than what we saw in the last paper, where transitions are these things that are different states, and then you want to capture the thing that sort of pushes it to a new state. So, you have these, you characterize the system, you characterize these phase transitions, and then that tells you what you need to know about the system. In this case, what they're saying is that traditionally, you look at what's going on inside the compartments, inside the cells, and then that tells you, like, if you can characterize those dynamics, you can determine what's going on with the embryo. Of course, that's what they're also saying, that that's an incomplete description. So these people are critiquing that view. Then, of course, you can couple genes and their regulation into a gene regulatory network. So each of these compartments have a GRN, or a gene regulatory network, that is informative and gives you that same output that you would get for a set of equations that would describe a phase transition. So, however, it has been challenging to understand how these components give rise to the dynamics of patterning, in part because the list of components is very likely far from complete, and the interactions between them remain uncertain. So when we build a genetic regulatory network, we usually pick certain genes. We don't understand fully how there are other downstream processes that affect, say, the synthesis of proteins, or the synthesis of signals that travel outside the cell. And there are a lot of things we don't have that are really precise. We don't really have precise knowledge of. Not so much the processes, but like the numbers that you put on them. However, it is also because these patterns emerge on timescales different from those corresponding to the underlying genetic activity. So the genetic activity occurs at one time scale, like microseconds to seconds, just in terms of the processes. Sometimes, like protein folding and protein synthesis, happens on a very quick time scale. But these other processes, like developmental, phase transitions or changes in state, symmetry breaking, those all occur at very different time scales, like on the time scale of days. And so this is a big difference in time scale. So this is something that needs to be filled with, there needs to be some link between the two time scales. So this paper has a lot of nice figures. It shows like traveling wave fronts within cell arrays. So this is where, you can change the state using signaling between cells. They show these different properties. They also show genetic oscillators. So if a genetic oscillator is driving this process of differentiation, it looks different than if you're getting signaling across cells. So this wavefront is where you have a signaling dependent interface, this arrow, and this tells you, this kind of mediates this kind of change where there's this striping that goes on. These two different stripes of colored, the cells have different colors as this wave goes through it. With a genetic oscillator, the single cell level is the thing that's driving change, so you get this sort of mosaic of different colored cells. And you get striping, but it looks a lot different. So there, depending on what you're claiming is the mechanism for change, gives you different outcomes. And this is a genetic timer where you have different kinds of striping, where the cell just changes state over time. So level of maturity determines its state. And so that can actually lead to striping in a different way. And so there are different mechanisms you can use to explain this, but it gives you different outcomes. I think if you're interested in this for like developmental agents, and I think we talked about developmental agents a couple weeks ago, this would be a nice model to apply to this, where you're looking at the cells, or you're looking at like the components of the agent, and you're like determining what they do by their state and by the change in the state over time. So this is very amenable to computation, this type of model. They also have things called phase waves, which are fast structures that move. They call them kinematic structures that emerge when the isolated cells of a single stable biochemical state. So these are things that, like scroll waves and things like that move through the embryo. So these are things where you have different levels of activity for this gene product, and it moves through the embryo like this. So there's this wavefront, and it's controlled by this circuit of genes. So this is, and then this shows you kind of how it works. It organizes things at that global level. So you're linking gene expression here in different cells with this thing that goes outside the cell, and coordinates the changes in state across cells, because it's going out into the cell, into the extracellular matrix, as they call it, and it's moving across as a front. So you have a lot of stuff produced over time by genes, on a very short time scale, and then there's accumulation, and then that accumulation contributes to this wavefront that moves across the cell, or across the embryo, and changes the state of each cell. So that's one way we can link those time scales. So there are a lot of different types of waves and wavefronts that get into these things here, different model organisms, and they also model molting in C. elegans, which is where C. elegans has like a exoskeleton that it molts over time. They just change their skin, basically. It's a cyclic process that occurs 4 times with a periodicity of 8 hours. So these are very regular patterns. These molting cycles are driven by some of these pattern formation mechanisms as well. So you can model a lot of things with these. You can model genetic timers where you have, where there's something set up by a gene expression network or a gene regulatory network that happens at regular intervals. You have these, you end up with an output like these oscillatory waves, but now it's a gene expression network that's responsible for this. You have temporal phase shifts and spatial phase shifts that are possible to model. So there's a lot of stuff in this that's not computational as a paper, but they kind of give you these models, these different state diagrams, and they show you how they map out in a graph. So you can use, this is very useful. A lot of this stuff in this paper can be used to model a lot of different outputs. So there are a lot of different things we can explore with that if people are interested. So I think that's it for today. I'm not going to go through that whole paper, but I just wanted to give people a taste of this sort of stuff. So, Shruti, did you have any questions about any of that? Or?
No, not yet. I thought the universal route data was interesting, but probably since I missed some of the things initially, so I could not get a hang of everything. But I guess I'll look into it with you and see more about it.
Okay, sounds good. I'll put the papers in the Slack and okay.
Yeah, that would be great.
Yeah. Okay, well, thank you for attending. Ujjwal and Akshay were here earlier, so thank you for attending. And see everyone around this week. Next week we'll be back to Monday, and we'll talk about Hacktoberfest more and setting up for that.
Yeah.
Yeah.
Yeah, that would be fun.
Yeah. Talk to you later. Have a good week.